So before we get into phosphorylations and phosphates, um, had a good question about how to study for the final exam. So what I'd say on that is review your online homework problems, review the exams, and then ask yourself. There's kind of three categories I would say that most students have. One is that you see that you remember the answer and you could explain it to somebody. So that's the first. If you can remember why that was the answer and you could explain it to somebody, then the truth is all you need is a light review of that. You don't need a lot. You remember it. It's now part of your knowledge base. If you look at the question, right, and you still know the answer, but you can't explain why, that's going to need some review, right? That you need to dive into that a little bit. And you need to figure out why that's going on, maybe look at the mechanism, look at the rationale behind it, but you're going to need to take some work on that. If you look at a question and you can't even remember where it came from, you don't know why you answered that, that's going to just assume that you've got to study that like you did your first time. Give it full attention, and really spend a lot of time. This is going to make your study for the final exam much more efficient. So I would go through all your old assignments, look at them, and see if you can figure out why you had the answers you had. And again, if you could still answer it and explain it to somebody, you're good. Don't spend a whole lot of time on that. If you are, you could answer it, but you don't know why, then you need to review that pretty extensively. And then if you really don't even know where it came from, you got to act like that was your first time. Okay. The quiz on Tuesday will be over the material we covered that wasn't on the exam. So that'll be both goes back to the epoxide ring openings and the thiols and the sulfides, and then a little bit of today's stuff. So phosphorylation. I will post an online homework for the phosphate information. So let's go ahead and get started with that. I've got a few images that I'll be bringing up from time to time. I'll post these notes in hard copy as well as video, so you don't have to, don't rush to try and copy down all of these pictures. So okay. So first, let's look at why are we looking at phosphates, and where do we have the important part of that. And when we look at phosphates, it turns out phosphates are right in the middle of DNA, right? So if we have some DNA strands, we can see that that's actually the backbone, right? So we have a phosphate, and then we have the deoxyribose. For RNA, we would have phosphate and then a ribose, right? So ribose. So the phosphates are going to be important there. We also know that we have ATP, and we'll be spending quite a little bit of time on with ATP today. ATP is referred to often as an energy molecule, right? So we have the triphosphate here. We've got a ribose sugar here, right? And then we have adenine. Okay. And so this ATP, ADP, and again, I'll post these so you don't worry about getting down these pictures real quick. So if we look at this one right now, this is just the diphosphate. Right, so really the only difference there is the diphosphate. So if we lose one phosphate group, and then the last one, AMP, right, and we just have the single monophosphate. And to stay with the MP, we can say mono. Okay. So biologically, we see phosphates in a few places. The other part of the phosphate, and the reason that it's good to go ahead and introduce it in organic, is that it's also related to a kind of a concept that we're doing in organic, which is activation to create a leaving group. So if we look at, activation of alcohols, if we wanted that group to leave, we could use an acid to get that group to be active, right? But this, of course, means that it's in situ, meaning that it's not like I can protonate the alcohol and then keep it there. Like, I'm not going to be able to isolate it, not going to be able to store it. It's really in the reaction and needs to be under acid conditions. So that one's pretty limited. The one that we looked at in organic is the tosyl chloride, right? We can make the sulfonate ester. And then that makes this a good leaving group, right? Because if we had this group as a leaving group, right, that's going to be delocalized, and we see that is going to be more stable. So we've seen those two activations of alcohols. Now the acid, that's going to be problematic physiologically because we don't have an acid solution, right? We have more or less a neutral solution, pH near 7, and so that's not going to be a good activating group. Within an enzyme, you can have protonation to form this. So it's plausible with an enzyme, but again, it's going to be a higher energy reaction. And biological systems, for the most part, are all based on really minor modifications of energy of activations. With the tosyl chloride is really reactive, and it's going to more or less react with whatever, right? So there are ways to control and make modifications to the tosyl chloride type reaction where you can specifically activate one alcohol over another. There's a lot of sugar chemistry. And again, if we think about a sugar molecule and why this is important is if we look at a sugar, right, we've got OH groups on every single position. So the tosylate's very reactive. The conditions are more organic than aqueous, a biological system. But we do have the possibility, and my sugar is off here. I'm not drawing it correctly. It should be oxygen here, OH, and then OH. So if we look at this one, right, we have all of these different positions. So 1, 2, 3, 4, 5, and then 6, right? So we have all of those different oxygen groups. And there has been ways through sugar chemistry to actually activate or protect each of those alcohols differently. So chemists came up with very ingenious ways to protect those. But in a biological system, this is all done with enzymes and ATP, So in a biological system, what we're doing is taking an alcohol, okay? And now we can think about it this two ways. The ATP, and the way this is usually shown, and we'll go over the mechanism in a second, is that you see a curved arrow on top to signify what's happening. So in this case, I'm going from ATP, right? So that's try, right? And I'm going to die, right? So therefore, if I'm going from try to die, then, and sometimes they don't show it, sometimes they will, but you know, this is usually enzyme activated. So this is biological systems, things work under a catalyst. Catalyst is the enzyme. So since this was ATP to ADP, then I'm going to end up with this phosphoester. Now this does two things. One, right, as I talked about here, this is activating, right? Because this is a very stable leaving group. So just like with the tosylate, the phosphoester is a great leaving group. So the first thing this does is it activates. The second thing you can do is do a temporary protection, right? Because I'm going from this proton to the phosphate. So I'm getting rid of that proton. So it does have a possible change in not the reactivity alcohol, but the reactivity of the acidic proton. Okay. You'll also see, sometimes you'll see something getting phosphorylated to enhance the way it works within an enzyme. So to make it fit with an enzyme, you'll see that that's not as common. Activation, in organic terms, activation is what we're seeing. Is we're seeing some type of addition of a phosphate group to an alcohol to actually increase its reactivity, its ability to leave. Sometimes you'll see it where it actually temporarily protects by removal of the hydrogen, but usually that's not major component. You'll see it in some biological mechanisms. And then the other is that it could be used to help with the phosphate. Now phosphates, let's talk a little bit more about just what we have as far as phosphate groups. So if we want to think about just generically what, just before we get into the mechanisms, let's just talk about it. So phosphoric acid, if we have enough time, I'll talk to you about the first isolation of phosphorus, which is quite interesting. So inorganic phosphate, this is usually abbreviated with a PI. So if you're looking at a biochemistry text or such, you'll see PI used quite a bit. Anytime you see PI, it means inorganic phosphate. That's just the way they refer to it. The reason that is referred to as inorganic is, notice there's no organic. So, you know, in biological systems, they're often attached to some type of organic group. So therefore, it's an organic phosphate versus an inorganic phosphate. The other one that's very common that you'll see is the diphosphate group, which is referred to not as a diphosphate, but as a pyrophosphate. And again, since this is not attached to the adenine, ribose adenine group, then this is known as organic pyrophosphate. And we'll see that one in mechanisms as well. So if you see a PPI, that's inorganic phosphate. Usually, if you see a P, that's phosphate. If you see PP, that's the pyrophosphate. And again, once you get into biochemistry, you're going to see quite a bit of abbreviations. There's lots of large molecules. They tend to use acronyms and abbreviations quite a bit. So if we look, at the phosphoric acid, we have 3 protons, right? So if we look at pKa, we're actually going to have pKa's. So if we think about the protonation, we have the phosphoric acid, and then we have 1 proton missing, so still 2 protons. And then the acidity of the species with 1 proton and 2 oxygen negatives, and then that results, of course, in the phosphate, or the inorganic phosphate in this terminology. So our pKa values here will give us what's happening in a physiological system. So if we look at the pKa of the first, this is a strong acid. pKa is 1. Okay, and usually, referred to as 1 or alpha. The pKa2 is going to be about 6.5. pKa3 is 13.0. And then, of course, the last one doesn't have an acid, so it doesn't have a pKa. So if we look at physiological pH, right? So 7, what that means is that slightly over 50%, right, is going to be in this group, right? So this is going to be the standard in physiological systems. So when we think about phosphoric acids and organics, this is really the one that we're going to be using at most of the time. Of course, in an enzyme, we can use an inorganic. We can have a protonation to get the inorganic phosphate. But physiologically, we're going to have about 50% of the H. Oh, I'm sorry. Let's see. No. Apologize. I drew the wrong one. It's going to be the H2PO4 minus. For phosphate esters, okay, and a phosphate ester is now we're adding some R group to our phosphorus. So if we look at this phosphate ester with the 2 R groups, so we'll have R1 and R prime. The R prime, I'm going to use that today quite a bit. We looked at it, I think, back in ethers, but just to remind you, the prime just means that this R is different than that R. It's just shorthand. So instead of saying, R1 and R2, we just use prime. This pKa is about 1, depending on what our different groups is. R, which means physiological pH. We end up with the anion. Okay. And it's the phosphate esters we're going to be spending most of the time discussing. This one is a diester. So the main mechanism here is going to be phosphate transfer. So if we have an R group and we react that with a R prime phosphate, the reaction we're looking at is going to be that transfer from one R group to the other. Oh, and let's go ahead and put the proton on there. So the base reaction is going to be switching these two. It can be done as this is shown with the R, again, referring back to biological systems. This is a biological reaction. We were really talking about ATP as our main phosphate transferase. Now, one of the things that's important in this is that most of these reactions are going to be promoted by manganese. Okay. So when we look at these, the reason this works is if we think about this phosphate, some attachment to some organic molecule, the manganese plus 2 can form coordination with these two oxygens. And so we refer to this as chelation, from the Latin for, I think it's Latin for crab, the basis, but chelation is, and you might have heard of that, certain amino acids are known for chelating. So therefore, if you've ever been on one of those diets or read about one of those diets about getting rid of heavy metals, they'll tell you, take this because it's a chelating agent. Chelation just is this idea of being attached to two different groups, kind of like a crab attaching itself with its pinchers. Okay, well, what this does, right, is that this creates then this more electrophilic phosphorus. So we can think about the manganese as increasing the electrophilicity of the phosphorus. And so manganese, so you'll see most of these enzymes are going to be that are used for the phosphate transfer are going to have magnesium. This is one of the reasons magnesium is an important part of our diets, right? We need to have the magnesium there. Magnesium is not hard to obtain through simple vegetables and stuff. Magnesium is quite common. It's less common in meats, but that's why sometimes you'll see manganese, especially on enzyme, metabolism based supplements. They'll have magnesium because magnesium is important for this phosphorylation. And if we remember biology, right, ATP is part of the energy system, mitochondria, and the manganese is important. That. Let me show you a couple of the images. And again, these will be up, so don't try and copy these down. So what would this look like with ATP, right? Depending on the enzyme, right? We could have one magnesium coordinated at this position, right? Between these two P's, phosphates, or we could potentially have it coordinated between these phosphorus. And there's also one where we could have it between the red and the green phosphorus. So when we look at ATP, you know, the phosphate, depending on the enzyme and the process that's going on, we have multiple ways where the magnesium can be coordinated. Okay. For simplicity, we're not going to add magnesium in as a coordination on all of these. You should think about when we say enzyme, that the enzyme may have magnesium there. The magnesium may be promoting this. So I'm not going to be showing it in the mechanisms for the phosphorylation. We're just going to be looking at the organic portions. We're not going to be looking at all of the incredibly large enzymes that would be doing this. But it is important to remember that many of the phosphorylations, especially with ATP, are going to be promoted with magnesium. And so when you get to biochemistry, you may see this. You may see the magnesium included more often. If we think about this mechanism, we have three variations of the mechanism that are still somewhat in dispute. The first one I'm going to show you is a concerted one, is the one that most people like. It replicates the SN2 mechanism, so this one is often called SN2-like mechanism. Easiest is going to be it's called concerted. But in this case, if we take our R group with the proton, and we have our base, and our base is going to be part of the enzyme. And we have our phosphate group, and again, this could be the base is going to deprotonate or do a proton transfer for the alcohol. That frees up these electrons to attack the phosphorus, and then the oxygen, the other ester, will leave. Right. The intermediate here and let me change this. So I'm going to show this as this species to make this intermediate more along the lines of what you would see. So if we have our oxygens here, now we got the partial bond here, partial bond there, our new R coming in, our old R prime going out, and so we end up with this transition state. And then that transition state results in now our new bond. Positive. Okay, and then this can react, and then this is in equilibrium with the double bonded oxygen species, right? So that's just I'm showing the zero ion, the separated zero ion, as the reactive species. Okay, so concerted mechanism in an enzyme, right? We can get the alcohol group exactly where we need it so that it's reacting with a proton acceptor, our base up here. That then reacts with our ATP, and we end up with our product. So now this is a pentavalent transition state. Okay. This mechanism has been studied. Now notice that the phosphorus has all oxygens, right? So with all oxygens, there's no way to really look and see what's happening with the stereochemistry. But the phosphorus, if the oxygens were different, would be chiral. So there were studies where they looked at this and actually made specific isotopes of the oxygen. So oxygen 17 and oxygen 18. And with those oxygens, they were able to verify that there is this type of inversion occurring. So if we think about it, if this was chiral, meaning the oxygens were different, we would see inversion. So this idea of taking, you know, this oxygen negative, but that one's a 17, and this oxygen negative, but this one is an oxygen 18, and this one's a 16, you know, and going from this group, right, and coming out with this group, right, they were able to show that this is inversion. So incredibly complex experiments with isotopes. Okay, so they showed the inversion, which does support the SN2-like, but it also does not exclude the other alternatives. So this is the mechanism I'm going to be showing most often. This is the mechanism that is favored right now. But the thing about science, and I think this is one of the things with the pandemic, that it has been hard for those that you know aren't fully versed in how science works, and that's a large population, even people that have graduated from college, is that science is always testing its hypothesis and testing things. So science can change. Things should never be really codified in science. There are some things that become laws, but they're very few, and they have to gravity. They have to be there for extended time. In this case, this really wasn't discovered until, it wasn't really kind of experimented on until the late 70s, early 80s, which is in my time scale. And still to this day, there's still people doing reaction mechanisms on this to understand how phosphorylation works in biological systems, and biological systems are complex. So minor changes in enzymes could have these. So just like with the COVID and the fact that as we learned more, different things changed. If we learned more about how long people are have to wait for the symptoms, so the one that just happened, right, is we went from 14 days down to 7 to 10 days, you know, finding out that the symptoms do occur in a much faster time period than originally thought. That's what the data is showing. So, you know, things change. And so in this case, I'm going to show the other alternatives. But the rest of the day, I'll show the concerted. So the other ones are possibilities; they're not excluded, but you know they're still being studied. So the first alternative mechanism is known as the addition elimination. So a two-step process. In the addition elimination, we're going to end up not with a pentavalent intermediate, but actually a pentavalent, not a pentavalent transition state, but a pentavalent intermediate. Again, we have this similar setup. And this time I'm going to show this resonance structure. So the bromine pulls off the oxygen here. Are y'all hearing music?
No.
No? Okay. Never mind. My kid might have left something on in the background, so just want to make sure it wasn't playing through my computer. Thank you for answering. So if we look here, then so what's happening here is that we get the base again pulling the proton, then the electrons coming around. Now in this case, instead of automatically kicking off our ester, we'll have the lone pair go up. That results in this intermediate. Okay. So in this case, it's not a transition state, it's actually an intermediate. So the idea of the addition elimination is in the first step, we have the addition. Change my colors. Right, and then in the second step, we have the elimination process. Okay. So the oxygen negative comes down, kicks off our group. We still, if we could study the stereochemistry here, again, with isotopes, it would be inversion. The only difference here is kind of our order of reaction, right? In this case, we're going to end up with an intermediate versus just one step. The next one is, so that one is the alternative at addition elimination reaction going through an intermediate. The next one is elimination addition. Okay, elimination addition is going to be SN1 like. In this case, we start again with our enzyme. Draw that too far over. Our phosphate. Oh, wait, never mind, sorry. Excuse me. Sorry to take up your paper. Okay, so in this case, we start with the phosphate ester. All right, so we've got our phosphate ester. And the first step, the oxygen leaves. This ends up giving us the phosphorus positive. So then this pulls that, and then that will react there. Again, what's going to be preferred, or at least what I'll be showing over and over again is, or what I'll be showing a couple more times, is the SN2 like, the concerted mechanism. Now at this point, it's important to say that whether the mechanism goes through SN2 like the concerted, the addition elimination, or the elimination addition, because there's no stereochemistry with the oxygens, they all give the same products. So this is distinctly different than SN1 and SN2 when we looked at carbons. With carbons, we can't go through the pentavalent. We often have chirality, and the stereo and regio chemistry of the SN1 and SN2 reactions of carbon give distinctly different products. So we need to be able to differentiate whether something is likely going to go through an SN1 or is likely going to go through an SN2 because it changes the products. With this phosphorylation, this phosphate transfer, all three of these really give about the same product, right? So we're not going to need to distinguish between these, and it's still disputed. So when I say SN2 like, the concerted, that's the one we're going to be using. Don't think that that's the simple one and that the SN1 will give us something different. The truth is that in this case, all three of these mechanisms could potentially be giving the same stereo and regio chemistry. So there's not a easy demarcation between these two. Okay, so I will not ask you to identify whether it's concerted addition elimination or elimination addition, but it is important to understand that, you know, science is still progressing. We're still learning things, but I'll be showing the SN2 like. Okay. So let's take another look at the adenosine triphosphate. And here we have the phosphorus. So let me pull up one of our an important part in the adenine phosphate is that we also have these three different phosphorus groups, and they actually have different names, starting with this phosphoester group. All right, so this is going to be our starting point, and so that means that then this one is going to be alpha. All right, this one's going to be beta, and then this one is gamma. All right, so we're going to refer to those three phosphorus with these terms. Again, starting at the ribose, right? Starting with that phosphoester bond, the alpha, beta, and gamma positions. If we have phosphorus attack at the gamma, okay, so the phosphorus gamma reacting, what's going to happen if we take our group, react that with ATP, let me change my colors back. You know, and it be promoted to do the gamma, what we'll end up with is the phosphate here. Plus then ADP, right? So we're going to end up with the diphosphate. If we have beta attack, so again, thinking about an alcohol that we're going to add a phosphate to, again, using our ATP, an enzyme, and again, these would be very specific to the conditions. So my enzyme with beta attack, what I'll end up with is, and the mechanisms on these are going to be, again, that concerted type mechanism. Here I'll end up with the pyrophosphate addition. And again, in physiological systems, we usually would show it with the oxygen negative, and then we end up with AMP, so the adenosine monophosphate. If we look at alpha, oh, that's a gamma, sorry. If we look at alpha addition, it's going to be quite different. So if we take our substrate here, we react that again with ATP, that's going to be our main phosphorylation group. Enzyme, right? But now the enzyme is made for alpha attack. In this case, we don't end up with a triphosphate. What we end up with is the actual phosphorus with the ribose A is the abbreviation. So the ribose would be all of this, okay. Usually abbreviated as ribose A. Okay. So we end up with that ribose A, and then what is then given off is the inorganic pyrophosphate. Questions about these? So with the ATP, we have three different phosphorus. Each one of those gives us a different type of reaction. Again, we'll be using the concerted type mechanism. So if the enzyme is set up to attack at the gamma, and again, you're not going to be given a structure. So I'm not going to give you, let's say, glucose and go from glucose 6 to glucose 6-phosphate, let's say glucose 6-phosphate, and ask you to determine what's going to happen. I would have to give you some indication, right? This is not, I can't give you ATP and enzyme without any other identifier and then ask you, is this going to be a gamma, a beta, or an alpha? Now I could show you the products and ask you if it had been an alpha, beta, or gamma, okay? So you're not going to predict whether it's alpha, beta, or gamma, but you should be able to recognize whether it's an alpha, beta, or gamma phosphate reaction. Let's just go ahead and do one. So I think I confused myself, so it probably confused you. So let's try a couple of these. So let's show this sugar, and this is a glucose. Okay, so we're going from glucose to glucose 6-phosphate. And again, the 6 from the nomenclature where that last carbon right there is 6. So this carbon being 1, this carbon being 6. Okay. So if I showed you this one and asked you which of the phosphates are being reacted or something along those lines, what's the reaction? Is this going to be a gamma phosphate reaction, a beta phosphate reaction, or an alpha phosphate reaction? right? So let's think about it. If it had been a gamma phosphate reaction, I only had a single phosphate, right? So I'm just adding one phosphate. If it had been a beta phosphate reaction, I'm actually adding a diphosphate. So one of the things that can happen, right, with a beta is instead of activating with one phosphate group, I can activate with two phosphates groups. And then with the alpha, I'm actually making the ribose anose here all the same. And where would you see this? Well, you would see this type of group when you're synthesizing DNA, right? Because there's the ribose. I just have to change this group here, and I can build up the backbone of DNA. So if we look at this one, right, what's happened? I've changed this hydrogen for a 1-phosphate. So the answer here would be that this is a gamma phosphate transfer from the ATP. Okay, so alcohol phosphorylations, phosphor transfers to get dupes. We also have phosphorylations not carboxylase. And now that we've shown it, I'll go ahead and use this ADP. So if I showed this, right, this is trying to get back to the idea of what type of questions you'll be seeing. Okay, if I was going to add a phosphate to this carboxylate, because I'm going from ATP to ADP, right, I can assume that this is going to be a gamma phosphor addition or transfer. And again, that's based on the fact that, you know, when you go from ATP to ADP, you're adding 1 phosphate. And again, the reason you would do this is activation and protection, modification of these groups, right? So once we add the phosphate, you haven't got the carboxylate chemistry yet. You'll see that in Organic 2 if you can continue on. In Organic 2, they'll talk about the reactions of activation of carboxylic acids, carboxylates. So this is similar to that. So this is going to be the phosphorus version, the biological version of activation of carboxylate. And similarly, we could add the diphosphate, right? So if we went from ATP to AMP, right, enzyme, we could end up with this. This would be phosphorus beta transfer. And if we went from ATP to the inorganic pyrophosphate, remember that's the PPI, then that would give us the phosphate with ribose A. Okay. Okay. Questions about these? I will post the learning objectives for these. Go over those quickly, and then we'll do some practice problems talk, and we got a little bit of time to talk about. So, what things you need to know? You need to be able to recognize phosphates, understand ATP, ADP. It's a multiple choice test for the final, so I'm not going to ask you to draw this, but you do need to recognize these. When you look at the reason for these, it is important to understand that this is an activation and a protection process. Do understand your different phosphates and organic phosphate, PPI. Again, if you get to biochemistry, you're going to see these all the time. If you're in bioengineering, you're going to see these all the time, PI and PPI. If you pKa's, understand the physiological pKa. I'm not going to ask you what the pKa's of each of these are, but you do want to understand what the pKa's mean as far as physiological pH. Same thing with phosphoesters. The phosphate transfer, you would need to recognize, you need to understand that the magnesium is involved. That's important. And you may be asked to show an SN2-like mechanism. So either identifying the intermediates or the transition state, pentavalent transition state. Less likely to show the alternative mechanisms. What you would need to know there is that they're all alternatives and that they are all in dispute. And then the ATP, you do need to understand the gamma, beta, alpha. This is very important. Understand the addition products of the gamma. You're adding one phosphate, right, to end up with ADP. For the beta attack or addition, you're ending up with two phosphates, right, plus the AMP, the monophosphate. And then for the alpha attack, we end up with a phosphate ribose adenosine. And then what's kicked off is the inorganic pyrophosphate. For phosphorylation of carboxylates, it really works about the same. The enzymes will be different, but the actual alpha, gamma, beta, and alpha attack, right, I didn't draw it there, will all be the same. Okay, any questions about phosphorylation? So our last topic is synthetic strategies. We've done some of these throughout, but let's go ahead and we're going to specifically look at multi-step synthesis and putting all the stuff we've learned together. This is a good way to prepare for the final exam. So when we look at synthetic strategies, you know, we have some straightforward reactions. So, but there's still a set of questions we should ask ourselves. So when I look at this conversion, what I need to think about is there any carbon chain? So one of the first questions I want to ask myself is, when I'm looking at synthetic strategies, is what's going on with the carbon skeleton? Have I extended the carbon skeleton? Right, well, so that means I'm adding, right, adding carbons. Have I lost carbons? have I rearranged carbons? If I've extended carbons, there's only a few ways we currently know, right? And this is part of kind of understanding the mechanism before we get fully into this. And we've looked at synthetic strategies on and off. What we're going to do is combine these all because this will help you review all of your chemistry. Is adding chemistry, adding carbons, right? We only know a few. So adding carbons, we know the we could do this one, or change that. Let me change my color too. Right, this one adds carbons. We've got our Grignard, the carbonyl. Right, that one adds carbons. We've got the Grignard with the epoxide. That one can add carbons. I think that may be exhaustive at this point. Okay. If we look at lost carbons, is there a way to lose carbons, like break a carbon-carbon bond? At this point, really, we don't know any, right? So we will get some, but loss of carbons is going to be a little bit more difficult. Think about it, it's more difficult because I'm going to be breaking a carbon bond. So there are ways to do it, but most of those will come in organic too. And then rearrangements, there's lots of organic rearrangement reactions. In fact, some very, very clever. But at this point, we're really talking about carbocations. All right, so we're talking about forming a carbocation and having that molecule rearrange. Okay, so I moved one of my carbons, so my carbon skeleton has changed. One of the second questions you need to ask yourself, and these aren't like you ask that question, you stop, you're going to ask all these questions, is functional group. Okay, and then, the question is, what is the starting functional group? And what is the ending functional group? And under here, there's several things to think about. One is, was it a substitution? Did I go from an alcohol to a bromide? That's a substitution. Was it an elimination? Right. In this case, it's not a 1-for-1, right? I'm eliminating that. Could it be an oxidation? Could it be a reduction? And in this case, we can see that there was no carbon skeleton, and it looks like it's been an oxidation, right? We're going from, we haven't substituted the oxygen is still there, but we have changed the oxidation state of the carbon. The third question I would, in this assortment, and it depends, you might jump all the way to 3, but I would ask all 4 of these, is regiochemistry, has there been some type of change? So let's say that we went from this to this, right? There's no carbon change, right? Our carbon skeleton is the same, so it's not a rearrangement of our carbon. We haven't actually changed our functional group, it's still bromide, but there has been a regiochemistry change. This turns out to be a few reactions to get this change to occur, but it's with the chemistry that you know at this point, this is very viable. So ask yourself about regiochemistry. And then the fourth one to always include is stereochemistry. So it's important as you review your reactions that you're always thinking about your regiochemistry and your stereochemistry. Okay, and with the stereochemistry, sometimes they can be stacked. I did that on the last test. It's one of the questions that I went ahead and gave you credit for. It was a very advanced problem. It actually took 2 stereochemistry changes to get the right answer. And so here, if we look at this one, right, there's stereochemistry involved. So whenever you start one of these synthetic reactions, right, you should be asking yourself these questions, right? At this point, you probably haven't been asking yourself these questions because really we've been doing one-step reaction. With the synthetic strategies that we're going to be looking at here at the end of the semester, we're going to be looking at 2, 3, 4, potentially 5-step reactions, right? How do we really get modifications to occur to change carbons? This is how we make pharmaceuticals. This is how we make synthetic molecules to create polymers, to create new materials. Is that we take known materials and we change them into something that has the properties that we want. So ask yourself the carbon skeleton question, right? Be specific. Did I add? Did I lose? Again, that's just counting. And then rearrangements, look at the overall structure. Then functional groups. When you look at functional groups, think about, was this a substitution? Was it an elimination? Was it an oxidation? Was it reduction? And also when you're studying your reactions, this is also a question you should be asking yourself, right? Did this reaction do a substitution? Did the reaction do an elimination? Did this reaction do oxidation, reduction? Regiochemistry, right? Always recognize your regiochemistry. Of course, with carbocation reactions, you have regiochemistry. You've got different regiochemistry for acid hydrolysis of an alkene to get an alcohol versus hydroboration oxidation. Versus potentially no rearrangement with the oxymercuration. So pay attention to your regiochemistry, and then stereochemistry, right? So you analyze. So the first thing you do with strategy is you should stop and analyze the problem, okay? This is a problem solving process, right? Think about it like word problems. Whenever you get word problems in mathematics, the tendency is to jump to just try and solve that problem as fast as possible. The first thing you need to do is analyze that problem. Are they using and? Well, and is plus, right? Does it look like groups? Well, that's a multiplication, right? So you need to analyze your word problems to actually figure out what strategy you need to take to answer it. That's what we need to be looking at on synthetic strategies. And so next Tuesday, when we come back, the quiz will be online. So I'll set it to take after class like we've been doing on the quizzes. It'll have multiple questions, so you won't all get the same questions. They'll be scrambled. It'll cover everything since the last exam, so that includes ring opening through phosphorylation. And then in class, what we'll do is we'll look at synthetic strategies and we'll just work several synthetic problems together. And then next Thursday, we'll look at a review for the final exam, which is on December 14th. And I forgot to look at the time for the final exam. And I have a question I forgot to answer. Oh, why is glucose good for energy storage? Well, sugars are good for energy storage from the standpoint of, if we look at glucose, right, we can draw glucose this way as well. So we can have an aldehyde there, and I can't remember which ones of these are there. If I was a good biochemist, and I'm not a biochemist, I would know all of these. So, okay. So glucose can be shown in these two different forms, right? The acyclic and the cyclic form, okay? Notice that in the cyclic form, I do not have an aldehyde, right? I have this group, right? So this would have been the aldehyde carbon. What does that mean? Well, I don't have an aldehyde, so therefore the aldehyde is protected. The conversion of sugars back to energy is actually going to give off carbon dioxide, right? So what's actually happening is you're chopping off one carbon at a time. And so as you chop off one carbon at a time, right, we get energy out of the process. As the aldehyde being protected, then we do not have that process. So when this is here, that stops this. So if I put glucose in this cyclic form, then I get it stored. If I hook a bunch of these together, so if I add another group here and another group here, then I get a starch, right? Or I get an amylose or an amylopectin, and you'll see that at the end of organic 2. But the reason that this structure is a storage, energy storage, is because I'm protecting that aldehyde. As the aldehyde, when this needs to be used, it is converted from the cyclic form to the non-cyclic form, and then the aldehyde and these carbons are progressively chopped off of each other. So glucose is the main sugar. If we notice this glucose, where have I drawn all of my OH groups? All of my OH groups are in the equatorial position. So that's why glucose is the most stable sugar. So you see glucose more because it's a more stable than galactose and the other ones, because all of the OH groups in glucose are in the equatorial position, not the axial position. So why glucose? Because it's the most stable sugar because of the conformations. Why is it good for storage? The cyclic versus the acyclic form and the protection of that aldehyde group. Okay, that's time today. We'll see y'all on Tuesday. Again, please get ready for your quiz. I'll put up some questions for additions to dienes and for phosphorylation. Okay, have a good day.